Hi everyone, my name is Katie. I'm an assistant scientist in the Wildlife and Ocean Health Program in the Anderson Cabot Center for Ocean Life at the New England Aquarium. In the first video of our series, we introduced our program and shared that much of our research is focused around measuring hormones in marine wildlife. Hormones are powerful messenger molecules that control different biological processes in the body, like reproduction, stress, and metabolism. By measuring hormones in marine wildlife, we can learn things about their inner health, including things we wouldn't be able to learn just by looking at them on the outside. One of the first questions we often get, though, is how do you even study the inner health of an animal that lives at sea? Or how do you collect a blood sample from a whale? The answer is, sometimes figuring out how to collect a sample is one of the most complicated parts of our project. It's also not possible to collect a blood sample from a large free-swimming whale, so we have to come up with other creative sample types in order to learn about these animals' hormones. Today, I'm going to take you on a tour of our ultra-low freezer, which is where we keep our samples preserved at a chilly -80 degrees Celsius or -112 degrees Fahrenheit, and tell you a little bit about the different, sometimes quirky sample types we use to study marine wildlife. The majority of our work is non-invasive, which means we typically collect the sample without ever touching the animal. Feces, which is the scientific name for poop, is one of the main samples that we study in our lab. Hormones end up in feces because after they have relayed their messages, the body breaks them down and ultimately excretes them, so a poop sample gives us insight into the recent hormone levels of the animal. Over 20 years ago, our lab became one of the first to study health in free-swimming whales, and that was possible because of feces. Although it might sound a little gross and can be a bit stinky, feces offers a treasure trove of information. In addition to the hormones we study, feces can also provide information about diet, disease states, genetics, and much more. The first species we studied and continue to study today is the endangered North Atlantic right whale. Right whale feces can be bright orange to red to brown based on their diet, and unlike some species of whale, it actually floats at the surface as a nice plop, making for easy scooping. The Anderson Cabot Center's Krause Marine Mammal Conservation Program collects these samples as part of their boat-based whale surveys, and then send them to us for analysis. It's always an exciting day in the lab when we get a new batch of fecal samples. In total, we have amassed over 400 right whale fecal samples across 20+ years, which are tucked away on the top shelf of our freezer for our ongoing research. The third and fourth shelves of our freezer are filled with fecal samples from animals that live here at the aquarium. In partnership with our marine mammal team, we are studying hormones in the northern fur seals here on site. Through our research, we're hoping to understand more about the complex reproductive patterns of northern fur seals. In turn, this information can be used to help provide the best care for our animals. A fun fact about this study, because there's multiple fur seals living in the same habitat, it can be difficult to know whose poop is whose. To solve this problem, our marine mammal team adds fecal markers to each seal's diet. Natural food items like sesame seeds and lentils are placed into the fish the seals eat, with each seal receiving a unique marker. These items are not fully digested, so the markers are visible in the feces and can help us match who the sample belongs to. When you picture a whale, you might imagine them coming to the surface of the water and releasing a big spray or plume of breath from their blowholes. Those big breaths actually contain very tiny amounts of hormones that we can measure. Dr. Liz Burgess, along with others on our Wildlife and Ocean Health Program, have been working to quantify hormones in North Atlantic right whale blow, which is a novel advancement for the field of whale conservation physiology. Hormones in breath are thought to reflect the near real-time concentrations of hormone in the whale, making these measurements particularly valuable to understanding the immediate impacts of possible disturbances or stressors on the whales. Interestingly, one of the first steps in our study of blow hormones was simply to determine the best way to collect the breath. Our scientists tested different collection devices which were suspended above the whale from a long pole. They tested materials like wadded-up bridal veil or mesh netting, but ultimately found the best collection vessel was a giant Petri plate-like dish. Blubber is a dense layer of fat that lies beneath the skin and is a unique characteristic of marine mammals. For a number of years now, the Krause Marine Mammal Conservation Program has collected small pieces of skin and underlying blubber from North Atlantic right whales using a specialized biopsy dart. These biopsies are used for invaluable genetic analyses critical to understanding the population. Leftover pieces of blubber are then placed in the freezer for safekeeping. Recently, our team conducted a study to measure hormones in these tiny pieces of blubber, giving us another tool to explore right whale physiology. Blubber is quite oily and can be challenging to work with, so another aspect of our study was flexing our laboratory skills to develop a safer and more efficient method of extracting hormones from blubber than used in past studies of blubber hormones in other marine species. When working with some marine species, it is possible to collect a blood sample, such as when we partner with the aquarium's animal health department to study the cold-stunned sea turtles that come through our rescue and rehabilitation center. In these clinical studies, we measure a panel of several different stress and metabolic-related hormones, which provide valuable data to track the changes in the sea turtles' health throughout their rehabilitation and before release back to their ocean home. I hope you've enjoyed exploring our freezer archives. Each different sample type we study gives us a unique picture of an animal's hormone patterns. The more techniques we develop, the more tools we have available to understand the impacts of a changing ocean environment on marine animal health.